You know, inside every one of our cells is this incredibly complex code, our DNA. And sometimes just a single mistake, a mutation, can be the spark that ignites cancer. For years, scientists have been grappling with this huge question. In this vast ocean of genetic code, which genes are the real troublemakers? Which ones, when they break, are actually driving the cancer?
Right, let's get right into it. This is the central mystery, especially for researchers studying complex diseases like B-cell lymphoma. If you can't figure out which genes are the true drivers, how can you ever hope to develop treatments that actually work? It's step one, and it's absolutely crucial.
But here's the thing, the problem wasn't getting solved, it was getting worse. As our technology for sequencing DNA got better and faster, the list of potential culprits just exploded. I mean, for just 3 types of B-cell lymphoma, scientists had a list of over 850 different candidate genes. That's a lot. And this flood of data, this deluge of drivers, turned out to be a real double-edged sword. It sounds great, right? More data? But it was actually creating more confusion than clarity. Why? Because a ton of these were probably just false positives, genomic noise, that was sending researchers on wild goose chases and really slowing down progress.
So you have to wonder, how did we even get here? How did we end up with this ridiculously long list? Well, let's take a look at how the number of these candidate genes just spiraled completely out of control.
Just look at this timeline for one type of lymphoma. In 2011, the list had 81 genes, totally manageable. But then, by 2013, boom, 456. And it just kept climbing. A decade later, it was pushing 700. It felt like every new study just threw more names into the hat.
And this created this really big problem called orphan genes. Think of it like this. A lab publishes a paper, gets all excited about a new discovery, but then no other research group can ever confirm it. It's just a one-off finding, a single data point floating in a sea of noise.
And this chart really shows you just how bad it got. I mean, look at study A. A whopping 82% of the genes they identified were orphans, never seen again in another study. Another one had over half. The scientific community was trying to build this incredible structure, but the foundation was, well, pretty shaky.
So, faced with all this chaos, one team of scientists basically said, okay, enough is enough. It was time for someone to step in, clean house, and become the world's first ever gene auditors.
This whole effort was led by the lab of Dr. Ryan Morin over at Simon Fraser University. And his mission, right in his own words, is all about cutting through that noise to find the real drivers, the genes that truly matter in B-cell lymphomas.
Now, the 2 lead authors who spearheaded this massive project were Dr. Krista Coyle, who's an expert in these repeating mutations, and Dr. Konstantin Drevel, a bioinformatics whiz. You couldn't have picked a better duo, really. Their combined skills were perfect for a problem that was 1 part biology and 1 part big, messy data. And their solution? It was so elegant and just so powerful. They came up with a system, a framework, to audit every single candidate gene ever reported and sort them based on how strong the evidence actually was. Okay, so here's how their process worked. Super logical. First, they collected every single candidate gene from every single study. Then, every gene started out in the middle, in Tier 2, which basically means, plausible, but we're not convinced yet. A gene only got promoted to the top, to Tier 1, if it had rock-solid evidence, like being confirmed in a totally separate study. And finally, genes got knocked down to Tier 3 if the original data just didn't pass muster. And this created this really simple, really clear system. Tier 1 genes, those are the real deal, the high-confidence drivers. Tier 2 are the maybes, they need more proof. And Tier 3, those are the ones researchers should probably just ignore. They're most likely false leads or just plain old data errors.
So after all that work, what was the result of this huge audit? Well, in one word, clarity. They took this absolute mess of data and turned it into something clean, reliable, and incredibly useful. So remember that list of over 850 genes? After their meticulous review, they boiled it all down to just 144 high-confidence Tier 1 drivers. Think about that. They basically cut through 80% of the noise to find the true signal. It's an incredible achievement.
And right away, this clean list gave us a critical insight. Look at this. There's a really significant overlap in the driver genes between these 3 different types of lymphoma. What does that tell us? It means they share common weaknesses, and that is a massive clue for developing therapies that could potentially work across multiple types of cancer.
Now, this wasn't just some boring academic exercise. Creating this definitive, high-confidence gene list has some profound, real-world consequences for cancer research and ultimately for patients.
What it does is provide a clear roadmap. Scientists can now focus their time and money on targets we know are real. It's going to stop labs from wasting millions on false leads. It means we can design way more accurate diagnostic panels for patients. And really, it just creates a new gold standard resource that's going to speed up the entire field.
I mean, here's a perfect example. Before this audit, trying to design a diagnostic panel was kind of a guessing game. The paper actually points out that some existing panels completely miss genes we now know are critical, like KMT2D in one lymphoma or CTSS in another. With this new list, you can avoid those kinds of huge mistakes.
But you know how it is in science. When you finally answer one huge question, it almost always reveals an even bigger, deeper one. All this newfound clarity has really just set the stage for the next great challenge.
Take a look at this. Even for the 144 confirmed Tier 1 genes, the ones we know for sure are the culprits, there's this giant gap in what we actually understand. For so many of them, we know they're involved, but we still have basically no idea how they're causing the cancer.
And the numbers here are pretty stark. The kinds of experiments that explain what a mutated gene actually does to a cell, well, they've only been published for 58 out of the 144 Tier 1 genes. That leaves this massive, uncharted territory just waiting to be explored.
So, at the end of the day, thanks to the incredible work of these gene auditors, we finally have it, the definitive list of culprits. And that means the next great challenge for a whole generation of scientists is crystal clear. It's time to go figure out all of their secrets.